Hi, I'm Judy Cole, the Executive Vice President and CEO of the MIT Alumni Association, and I'm delighted to welcome you to this web production of the MIT Alumni Association.
Please welcome to the stage Jacquin Niles.
Good morning, and it's really a pleasure to be here this morning to share with you some of the work we've been doing in my lab, where we work with the malarial parasite Plasmodium falciparum and devising strategies to use engineered parasites to really inform drug discovery applications to treat malaria. So just to give you a bit of context for this disease, malaria has been eliminated in most of the developed world, so including the United States. Until the 1940s or so, malaria was an endemic disease. It's been eliminated in most of the developed world, but continues to be a devastating problem in the developing world. So there are more than 200 million cases of malaria each year, mostly in the African continent and Southeast Asia. This disease causes about 500,000 deaths per year. And in the context of modern medicine and what we're capable of doing, this is still an unacceptably high number of people. Again, the most devastation we see is in the developing world, in the African region. There are 5 different parasite species that cause malaria in humans. The 2 most common that you've most likely heard of are Plasmodium falciparum and Plasmodium vivax. And Plasmodium falciparum is the species that's most dominant in the African continent and that's associated with the most severe forms of malaria. So most of the deaths that are recorded in that 500,000 cases, deaths from malaria, are mostly due to Plasmodium falciparum. And so this is the particular species that we've been trying to develop technologies in the lab to better aid our ability to treat, manage, and prevent, and hopefully in the long run, eliminate malarial disease. So to give you some context for what this organism's biology is, and in principle, why it's been so difficult to eliminate, especially in the developing world, we can think through the life cycle of these parasites. So it's a mosquito-borne disease. So parasites are transmitted to humans by the bite of the mosquito. These parasites injected in the skin travel first to the liver, where they undergo a rapid development process over the course of about 2 weeks. A single invading parasite might replicate to about 10,000 new parasites. At this point, the person is completely asymptomatic. After that period, parasites can rupture from the liver and exit into the bloodstream. And these parasitic forms are then capable of invading red cells, where they can then undergo a replication process. So a single parasite invading a red cell at the end of some developmental period can produce 20 to 32 new parasites, which then rupture from a red cell to be released and to repeat this process over and over again. And so untreated, you can imagine how a single parasite inoculated by a mosquito can rapidly lead to huge numbers of parasites within the bloodstream of an infected person. And all of the symptoms that we associate with malaria, all of the mortality and morbidity, are really due to this blood stage of the disease where parasites are rapidly replicating and overwhelming our defensive mechanisms. At some small frequency, we don't understand why, but these parasites will transition into sexual forms, which are then taken up by another mosquito, where the parasite will complete its development within the gut of the mosquito. So the challenge here is that given some of the biology of malarial parasites, they change their surfaces very rapidly, and they always stay one step ahead of the immune system. It's been very difficult to have an efficacious vaccine against malaria. So to this date, the best vaccine that's been most farthest advanced still offers only about 20% protection, and that wanes over time. And so what that's meant is we've relied on a number of other interventions to manage and treat this disease along the way. So you can imagine, given the life cycle I've described, that insecticides to control mosquito populations have been an important strategy in managing malaria. Mosquito nets, simple technology, but works very effectively at placing a barrier between a person and a mosquito, have been quite effective in blocking transmission as well. But invariably, even if you're religiously using insecticides or mosquito nets, people get malaria. And so the gold standard today is to treat them with anti-malarial drugs. So for the longest time, we could use chloroquine as single monotherapy. It's cheap, probably about 25 cents or 75 cents could cure you of malaria 20, 30 years ago. Today, this is not the case anymore, and resistance to chloroquine has spread quite rapidly. The WHO, World Health Organization, recommendation now is to use artemisinin combination therapies to treat malaria. And these started out being extremely effective in eliminating parasites from infected persons. But again, what we're seeing is this emerging pattern of resistance to the artemisinin-based combination therapies. So beginning in the 1940s, for example, with chloroquine, we saw resistance emerge first in Southeast Asia and then spread globally. So now every endemic area there's chloroquine resistance. And what we're seeing emerging with the case of artemisinin combination therapies is that, again, in Southeast Asian countries, we're seeing the emergence of what looks like the precursor to full-on resistance. So treatment failures at day 28 are starting to rise pretty significantly in these regions. So what the challenge then is, in the context of no approved vaccine and failing first-line anti-malarial drugs, we now need new strategies to think about both vaccination and drug discovery efforts. And so what I'll share with you today are some of the approaches that we're using to think through the process of how we can validate biological targets as potential points for drug discovery going forward. And then to think through how we might be able to devise strategies for rapidly identifying lead compounds that target these biologically validated targets within parasites to then advance drug discovery efforts. And so the types of things that we do, just in overview, are to take publicly available genomics data. So the genome of most of these parasites have been sequenced. So we have blueprints of what their genetic composition is. We have a sense of which genes parasites' genomes encode. What we don't have is any sense of how functionally they're used by the parasite, either to survive or to cause disease. So we can begin with these types of genomic level information, then build out sets of technologies that allow us to go into parasites and manipulate when particular genes are expressed, to then relate that to their biological functions, to then take those types of functional tools to then thinking through how we can do phenotypic screens that would allow us to understand which compounds available through chemical libraries that are available that could then target specific genes and give us a sense of what might be lead anti-malarial compounds to develop further. And through this process, we're very cognizant of the idea that the notion that very few companies are invested in drug discovery and development in this family of organisms. And so it's really important to think of efficiency with which we can take a lead compound through the developmental pipeline to then implement downstream. So the way that we've been thinking through this problem then is since we know exactly what the genetic composition of parasites are, can we create these types of resources in which we can interrogate individual genes and ask whether they're important to some aspect of parasite biology or to disease? And so the first level then is to create genetically modified parasites that inform us of the importance of particular targets. We can use that information then to prioritize which targets might be interesting to advance to some kind of clinically important application. We can then take and screen these lines against anti-malarial compound libraries to then very rapidly map which compounds interact with which targets. And so in a standard way, this would be done by taking parasites, screening them against a compound library. We can do phenotypic screens extremely quickly within parasites. In 3 to 5 days, we can know a given compound kills parasites. But then in the standard approach, we have this lag period of months before we can understand why a particular compound kills parasites. And what that introduces is an inefficiency with respect to how we can leverage the technologies that we are developing to inform drug discovery. So the approach that we've been working on is to see how we can go about using phenotypic screens with target information embedded within the phenotypic screen. So exactly within the same time frame of executing a screen to know we have an effective compound, we simultaneously have information as to which target that compound is most likely interacting with. And what that does is it increases the efficiency with which you can integrate the biology, medicinal chemistry, and other ancillary services with the drug discovery and lead optimization process. So how this works then is we develop these technologies. We can embed these technologies using the type of CRISPR, Cas9 genome engineering strategies you heard about earlier, to then code ways for controlling when a particular gene is either turned on or when its expression is turned off. And when we implement these types of technologies, we can get very good quantitative dynamic ranges between when a gene is turned off versus turned on. And we can start prioritizing different parasite targets into things that are most likely essential for parasite survival. So the technology then works to inform us of which things should be prioritized for advancing to drug discovery type efforts. So here's an example of how we are using this technology. So we have a set of compounds. We think they might target a given gene and not other genes. How do we use the ability to tune the expression of the gene to give us a sense of whether that compound is killing parasites through a particular target? And it's a simple idea in that if we think that a compound kills through interacting with a particular gene, if we turn down the expression of that gene, we make parasites more susceptible to the effects of that particular compound. And simply then, by mapping out these sensitivity relationships, we can understand the relationship between a compound and a target gene. So as an example of how we've been using this, the family of proteins that have been people are getting really excited about as anti-malarial targets are the tRNA synthase enzymes within parasites. This is how cells make proteins to help them survive and do biological functions. And what we can do with this then is we can modify the family of tRNA synthases within parasites, and we can either through short-term assays see which of these targets have very dramatic growth defects in parasites, and in longer-term assays understand where there might be fitness costs associated with certain targets that emerge over time. And so what this collectively gives us is a sense of which particular targets might be important or interesting to prioritize for downstream studies. So we can then take and ask whether particular targets interact with compounds that have anti-malarial activity. And in this case, I'm showing you a drug that kills parasites through this mechanism and not these other mechanisms. And similarly, another compound that kills parasites through a different target, as shown here. And so when we map out these relationships, what we see is as we titrate the expression of our target gene, we can see parasites becoming hypersensitive to these compounds. And what that tells us very quickly is that these are likely the cognate gene compound interactions that would then implicate this as a target, this protein as a target of that particular compound. This approach is specific, so we can screen different target genes, and only when we are looking at the cognate interaction between a compound and its target gene do we see a signature of a relationship between those two proteins and their compounds. And similarly, for a different target, we can map these types of relationships quite well. And so with this, what we're doing then is starting to move out into the space of thinking through how we could use these types of approaches to solve a very realistic challenge, which is how do you go from optimizing a lead compound, generating variety around it, while understanding that you're hitting exactly the same target that you're interested in. And we've done this with a particular family of analogs that are known inhibitors of a really exciting family of compounds that are targeting and really potent antimalarial activity. And again, what we can see here is we can explore now analog space to really quickly map these types of interactions with the target compound. And so in summary, what we've done then is started to create this expanded library of targets. And what we're starting to do is screen them against a larger set of both public and private collections of compounds with the idea that within 3 to 5 days, we can very rapidly glean information about antimalarial compounds and the targets to advance their development towards preclinical studies. So I'll end here by thanking the folks who've been working extremely hard to do all of this work. What I described is work done mostly by Shumanta Day and Sebastian Smick, but the entire lab's effort has really contributed to this work. And I'd also like to thank funding sources. The Gates Foundation, in particular, has been a champion of work in malarial diseases, and much of this would not be possible without that investment. Thank you for your time.
Thanks again for joining us. For more information on future MIT Alumni Association productions, please visit our website.